Dr. Baliga here. This podcast belongs to a group of 10 podcasts on statistics in medicine. These podcasts should give you a solid foundation on this topic. It's derived from an outstanding chapter titled Statistics in Medicine in Baliga's textbook of internal medicine, available at www.mastermedfacts.com. The chapter is authored by Dr. Donna Windish, MD, Associate Professor at Yale University School of Medicine. She is the Associate Program Director for the Yale Primary Care Internal Medicine Residency Program. Dr. Windish completed her medical school degree at the University of Connecticut and her internship and residency in New York at the University of Rochester. She went on to do fellowship in general internal medicine at Johns Hopkins University School of Medicine and obtained her MPH at Johns Hopkins Bloomberg School of Public Health. Dr. Windish has been on faculty at Yale since her fellowship. She is clinically active in both inpatient and outpatient medicine, oversees the research and residency program for the Yale Primary Care Training Program, and teaches medical students and residents biostatistics and how to read medical literature. Multiple choice question. Researchers designed a study looking at cancer deaths comparing a new chemotherapeutic drug to standard of care. They determined that they would need 80 patients in each group to detect a 20% difference in mortality given 90% power and a significance level of 0.01. Which of the following changes would require the researchers to increase their sample size? A. Aim to detect a difference of 20%. B. Specify a power of 80%. C. Use a significance level of 0.05. And D. Aim to detect a difference of 10%. And the answer is D. Aim to detect a difference of 10%. In order to see a smaller difference between a group, one needs to increase the sample size of the study. Increasing study power would also increase the sample size needed. Sample size. Researchers need to know prior to starting a study how many people will be needed to achieve a desired result. If the study has a negative result, it may be because the sample size was insufficient. You must remember, when evaluating the study, you must read the entire study, both methods and results, to see if the numbers of participants who remain in the study will meet the sample size limits when assessing statistical significance. As studies are being conducted on the COVID vaccine, one of the myths is the threshold myth. There is an incorrect presumption that there is a sample size below which the study is doomed. In reality, small sample sizes can have scientific merit, even if they do not meet conventional requirements for statistical power, and a valid sample size choices can be made for cost or feasibility reasons alone. Conventional power calculations provide precise sample sizes, but only by using precise assumptions. Accurately specifying such precise assumptions is challenging for any study and almost impossible for early clinical studies. Because small differences in assumptions produce large differences in resulting calculated sample size, uncertainty about assumed values makes it difficult for the conventional approach to guide sample size choices. Furthermore, one cannot ignore the cost of the pandemic and the feasibility of studies and the cost of conducting studies. Such conditions are often the real determinants of sample size. Early translational studies tend to have a high per subject cost and may put human subjects at considerable risk or require sacrifice of non-human primates, conditions that make large studies impractical and potentially unethical. Lastly, it is not possible to predict what will happen in a first-time intervention, so focus on any one specific hypothesis as required for conventional power calculations might not adequately reflect the potential value of the study. Alternatives have been recommended to the power-based approach for choosing and justifying sample sizes for early studies of new ideas. An established alternative in the statistical literature is the value of information approach, but this has rarely been used in practice. It calculates both the cost and value that can be expected over a range of sample sizes, choosing the one that maximizes value minus cost. The methods, however, may be too complex for frequent use with early studies. A more recent proposal is to examine 9 possibilities for the estimated effect and confidence interval that a study may produce. These possibilities result from 3 different possible effect sizes, that is, one that is expected or hoped for, no effect, and a possibility in between, and 3 levels of background uncertainty, including best guess, high uncertainty, and low uncertainty. A proposal then discusses how valuable the study would be under each possibilities. It has been argued enforcement of the requirement for at least 80% power has no valid justification and is especially inappropriate for early translational studies investigating new ideas. Practical considerations, including costs of larger versus smaller sample sizes, inevitably drives sample size choices, and the reality of diminishing marginal returns implies that this is scientifically valid. Indeed, the use of n root, determined only by cost, is reasonable for innovative studies. Say Peter Baccetti, Stephen Deeks, and Joseph McCune in an article titled Breaking Free of Sample Size Dogma to Perform Innovative Translational Research. It's published in Science Translational Medicine, 15 June 2011, Volume 3, Issue 87, pages 87. This podcast on statistics in Journal Internal Medicine is derived from an outstanding chapter on this topic in Baliga's textbook of internal medicine, available at www.mastermedfacts.com. It's authored by Dr. Donna Windish, Associate Professor in Internal Medicine at Yale University School of Medicine.